Hey guys, how you doing? Have an exciting update today. We have Lloyd Pye with us, and he's going to be talking about partial analysis of the DNA that was done on the Starchild skull that reinforces the absolute proof that can be obtained that the Starchild skull is not human. So let's get right to it. Welcome, Lloyd.
Hi, thank you very much. It's good to be with you again, Scott, and I'm glad to be with your audience again to bring them this update about the Starchild skull DNA analysis.
Okay, so you've just recently found out some more extensive information on the DNA. Can you drill down a bit and tell us what the process was?
Well, the process all along has been for our geneticists to find some piece, some fragment within nuclear DNA of the Starchild that has some meaning. Now, let me explain how it works. In all of our cells in the nucleus, we have all of the genetic material that our mother gives us and all that our father gives us. That's 3+ billion. It's over 3 billion with a B, billion base pairs. It's an awful lot of base pairs. So out of that, about 95% of the 3 billion doesn't really work. It doesn't do anything that anybody understands at this point. And when I say it doesn't work, they call it junk DNA. Everybody's heard it. They call it non-coding, non-functional, or junk DNA. Everybody knows that term. In that, if you pull out a fragment in the junk, it's just not. You can't tell what it does. It has no purpose. But in the 5% that works, if you can pull something out of there, you can compare what the proteins are, what the proteins make. And so the geneticists came to refer to that area as getting a fragment out of there as the golden needle. The golden needle in the giant 3 billion base pair haystack. Mm-hmm. If we could just find some base pairs strung together into a fragment there, and then he would be able to do something with it and make it a valid comparison against the human fragment of similar nature. So he's recovered dozens and dozens from the 95%, which were essentially not useless, but all they could do is run them through the NIH database, and it comes back either as recognized or not recognized in the database. Now, that was significant because when you have a lot of stuff that comes back not recognized, that's an indication it's not found on Earth. So that was significant in its own right, but not overwhelming. With the mitochondrial DNA recovery, it was the same way. He recovered 4 fragments out of the mitochondrial DNA, which amounted to 9.5%, almost 10%, which indicated strongly that the Starchild's the number of mutations in the mitochondrial DNA were going to put it well away from humans. Humans have about 120 mutations maximum in their mitochondrial DNA. Starchild's going to be in the range of 800 to 1,000.
Chimps have about 1,200, right? 15.
Chimps have 1,500, right? Yeah. So it's way away from humans. Not to chimps, but still no way you can call that human. Or even close. Neanderthal has 200. We have 120. Neanderthal has 200. Denisova, the new one, Denisova has 385. So, that was significant too, but not overwhelming. The new one is overwhelming. He got a piece out of the working 5%, and that piece is called FOXP2,
right.
The FOXP2 gene. Now, the significance of that is it's not just a functioning gene. It's a master gene that controls at least 300 other genes in what's called the downstream flow. In other words, what the FOXP2 does flows down to 300 other genes in our bodies. It controls them. It is a master gene. So what the geneticist said was, it's not just a golden needle, it's a diamond needle.
No, so it's a very big deal.
Very big deal. So the fragment that he got was 211 of the 2,500, roughly 2,500 base pairs in the FOXP2 gene, all right? 211. Not a big fragment per se, but it laps over something very significant in the gene, which is a couple of peptide strings. So it's unmistakable that this comes from the Starchild's FOXP2 area and that the Starchild has a FOXP2 area. But understand that just virtually everything that's of a complex nature, of complex life, and we're talking down to the smallest little animals, has a FOXP2 corollary of its own. Why? Because it controls basically the formation as your body is being formed, of your brain, of your throat, of your heart, of your lungs, of your guts, like the center part of your body. Some of the most important parts, not your limbs, but those core functioning parts. FOXP2 has a role in that, and it seems to have a role like that in everything. So it's very, very important, and it's very, very highly conserved. And highly conserved means is no changes allowed. Because if you have a change in the FOXP2, if you have a mutation in the FOXP2, it's going to kill you. If you're born, if you're being gestated with something goes wrong, mutates wrong, you're going to die probably in your mother's womb.
It'll abort.
Abort, exactly. So there's only a very, very few mutations known in this gene. One of the most famous ones, one of the best known ones, is if in the 2,500 base pairs, if you have one base pair different, you lose all capacity to speak ever. All right? It's that kind of deal. Now, with normal humans, Scott, and this is the key for everybody to understand, with normal humans, we all have exactly the same array of base pairs in our FOXP2 gene. The 2,500 plus that we're talking about, all the same, exactly the same. With the Starchild, there are as many as 600 to 700 if you project out from the fragment. The fragment itself has 56. A monkey has 2. A rhesus monkey has 2. A frog has 27. We have several animals outlined in the essay on the website. The point is, maximum for normal animals on Earth is 30 or less. The starch in that 211 base pair fragment. In the Starchild, it's 56. If you extrapolate that out to the full 100%, it's going to fall in the range of 600 to 700.
Because it was a partial.
Exactly. It's a partial. So the point is, there's no way to say this is a human, even close to a human creature, the Starchild. And now, we cover this base on in the essay that's on the website, the new essay that's on the website explaining all this, is that if a skeptic were to say, well, then it must be some kind of pseudogene, because it's so different, they're going to try to say, well, maybe it's a pseudogene and that's it. Now, pseudogene is genes that we do have in our bodies. Remember in that junk part of it, there are parts in the junk that seem to be genes that might have worked at one time, but they have been shut down for one reason or another and they don't work anymore. That's a pseudogene, basically. So they're going to say, well, okay, it's so different, it's so bizarre, it's so weird, it must be a pseudogene. Okay, fine, except there are no pseudogenes in the human FOXP2 gene, no pseudogene. So if you attempt to say it's a pseudogene, you're right away saying it's not a human. So we have them boxed in on that one. You see what I'm saying? They don't have anywhere to move. They either have to accept it as radically different from human, or they have to call it a pseudogene, which ipso facto means it's radically different from human. So we're in the catbird seat here, Scott. We're in really, really good shape now. Not with science. Science is still going to completely ignore us. They're going to ignore us until we can produce the whole genome, and every bit of it stem to stern shows that it's not human, which we know it's going to do. But this should put us in much better shape as we try to speak to investors to try to get the funding that we need, the large amount of money that we need to recover the genome and create the 2 documentary films that are going to cover the two major parts of what that entails, the recovery of the mitochondrial DNA and the recovery of the nuclear DNA. So if everybody can just be, patient a little while longer. We've been patient for 13 years. I hope that everyone that's listening can be patient for a little while longer. But this is a very, very major development for us. We're very excited about it.
Yeah.
I feel like this is the one thing we've been needing to put us over the top, and just have to continue on with what we're doing.
I figured it was when I read the update. I figured, oh, this is a big deal. I should probably talk to Lloyd about this.
Right.
How can you be sure that the genetic sample has not been contaminated in some way?
Well, it's just contaminated by what? I mean, what is going to, okay, it's contaminated by an alien because it's so different, Scott, what we have, but it isn't contaminated because it is a piece of FOXP2. It is a piece of FOXP2, and not only that, it is a piece of human FOXP2 because it has those two peptides. And what they are, basically, they're called polyglutamine tracks. I don't want to confuse anybody. But if you look, and it's on the website, you can see the string. We have the 211 base pair string compared to a few other animals, so you get a range of how that compare to their FOXP2 in that 211 base pair string. And, of course, you see a human as well. And what you see is in the human, there is 40 straight glycines. You'll see GLN, you'll see that, 40 straight of them. Then there's 6 different ones, and then there's another string of 10 more glycines in a row. And these are amino acids, and you have to be looking at that diagram to understand what I'm saying, but it's there. You can see it for yourself.
It's on your
well,
update.
those things, those same exact stretches are found in the Starchild fragment that we have, but the Starchild, instead of having what the human has in the other amino acid positions, it has a lot of glycines as well, extra more, and then the first 4 amino acids going into that stretch of glycines, and the 3 that are left coming out are all different. And you just don't have differences in humans. So everything about this says that it's coming from the Starchild's FOXP2, but everything about it also says it's not like anything approaching a human. So we feel like this is very strong, though partial. Everybody needs to understand partial, but incredibly strong partial evidence indicating that all we have to do is recover the whole genome, and we're going to be looking at an alien genome.
Okay, that's great. How much would it cost for DNA to be sequenced by, you mentioned this in your article, the next generation in the plank?
It's going to be a couple $1 million at least. Now, here's why. Everybody sends me these emails. Every time there's a new announcement about an improvement in the sequencing and a drop in the cost of sequencing, it's down now. You hear some new next generation builders, people who build machines in the next generation, saying it's going to be down to $1,000 before long. Well, that's true. And it's all applying to live, living DNA, my DNA, your DNA,
right.
A living body's DNA. Fine, that's great, but that's when all the base pair, all the genes, everything is strung together properly, and so the machines can just go down the line and pull out the base pairs and read them, and everything's great. But unfortunately, the Starchild remains dead. The old Chevy Chase thing. He's still dead. The dictator of Spain's still dead. Starchild's still been dead for 900 plus years.
Okay, so that answers my next question, was how long has it been around?
Yeah, its DNA is very, very badly degraded, very, very badly contaminated, it's ancient DNA. It's called ancient DNA. Anything over 50 years technically qualifies as ancient DNA. And so the reason that it's going to cost so much is what you have to do is make those runs through those machines literally dozens of times, so that you pick up, in one run you'll pick up X number of fragments. In another run you'll pick up X number of fragments. Won't always be the same amount of fragments. In another run you'll pick up another X amount of fragments. Again, some will be in the first 2, some won't be in the first 2. You have to do this dozens of times until you fill every hole. And that's what takes the time, and that's what takes the money. And so it's unavoidable.
So it's a multiple.
But, yeah, it's in the end, what you do, Scott, is you take, let's just say 30 for an example, because that's what they're tending to do now with the samples that they're doing of Neanderthals and Denisovans and things like that. So let's say they're 30. We're looking at 30 runs of the Starchild through the next generation machines, whether it's 454, whether it's Illumina, whether it's Xeon Proton, or whatever. And then you lay those 30 down, 30 lines on a row, and then you squeeze them down. You compress them until you you fill in every hole that you can in that genome. And then you're looking at one line with everything pretty much accounted for. And that's where that's when you say, okay, here's what we have, and here's where it's different from humans. And the geneticist has been saying from the very beginning when he got first results, he says it looks like it's going to be between 50% and 10% difference from the human genome. Now, understand what that means. A chimp is about 2 to 3% different. 97 to 98% of a chimp genome is the same as ours. 95% of a gorilla genome is the same as ours. So understand what this means. The Starchild, he thinks, is going to fall in the range of 10% to 50% away from us. Now, if you're that different from a chimp or a gorilla in your whole genome, there's no way to call that any kind of an aberrant human. That's an alien. Right. So, people say, well, how can you prove it's alien if you don't have an alien genome to compare it to? We're going to establish the alien genome to compare things to in the future. Compare the coneheads to. That's very important. The coneheads are out there. They don't look human, and it's going to be very important to have that as a baseline when it comes time to start doing this to the coneheads. So we're very excited about it.
Yeah, I'd be excited to see what the conehead DNA comes up as.
It's going to be much closer to human DNA, I can tell you right now, because the coneheads have much more human-like faces than does the Starchild, which has a very different, it's kind of human-ish, but it isn't human-like, really.
Right.
Like the coneheads are. They're different. They're clearly different. They have those brains twice the size of ours. They have some very significant differences in the jawline, etc. But I think in the end, the coneheads are going to fall closer to human than will the Starchild. The Starchild is going to be, the reason it's such a good thing for the Starchild to go first is because it's going to be so far away, there's just going to be no argument.
I remember years ago when we were emailing back and forth, there was maybe the mother was alien, or maybe the mother was human and the father
right.
was alien,
right.
And it sort of has been bouncing around over the years, but now it's looking more towards both parents being
alien. Exactly.
Alien.
No question. Yeah, for a long time, we had to go on the result of the 2003 DNA test, which indicated that the mother was human and the father was not.
I remember that.
But the test in 2003, the test still, you could only use primers. Now, with the next generation, with the new stuff that pulls out base pair by base pair, you get a much more accurate read, and we find out that the test in 2003, while accurate for its time, it was not completely accurate, not as accurate as it could be, and so it while we believed it for a long time that it was a hybrid, it really isn't. It's all the way alien.
Yeah. Okay. So very significant update. We know that we got a pretty good idea that it was the Starchild was around 900 years ago.
Right.
So we're not talking super ancient like 10,000 years ago or anything.
No degree of fossilization in the bone or anything. It's just bone and really, all things considered, for something dead that long, it's reasonably well preserved because it was buried in a mine tunnel. So it isn't,
And that helped.
Oh, a lot, absolutely.
So the samples that they're getting are good samples.
Very, very good for ancient DNA. They're good, clean samples. Exactly.
Okay, great. Can you just give everybody your website where you've got the latest updates? So
for those who want to read the new essay up that's about this, it's on the StarchildProject.com website, www.starchildproject, all one word, .com. And when you go there to the home page, you'll see a grid of icons to the right. The second one from the top says DNA and in brackets new DNA new. Click on that and you'll go right to the essay. Now the essay is 4,000 words. It'll take you about 20 minutes, 15 to 20 minutes, depending on how fast you read. It's got several good illustrations that make clear what's happening, but particularly you need to check out the the FOXP2 laid out against the human and the other animals, so you can see for yourself the importance of this and differences in the Starchild versus the human. You'll see the human stretched out in there in all blue, and you'll see the Starchild differences in red, and 56 are red out of 211. If it was 212, if there had been one more base pair that was the same as the human and 56 differences, it would be exactly 50% different. You see what I mean? So when he said 10 to 50%, when we saw that number, because we always believed it would be up closer to the 10% than the 50%, but just this little fragment of FOXP2 indicates it may be closer to 50% difference. So it's astounding, Scott. It's just astounding.
Yeah, it's a big deal. It's definitely, I've been following this, for a long time, and it's a very big deal that this has happened. So what's the next step? I mean, I got to ask, is the technician that you have doing the lab work, the person who's made themselves available to do this, are they doing it pro bono for you, or are they just sort of...
Pretty much. When it gets really expensive, we get we have donations that come in pretty steadily, and we apply those to it. But he does his time is all his own. We just simply pay for the reagents and the chemicals, which are expensive to do this kind of stuff. We pay for those, but other than that, he does it on his own time, generally on weekends, and a lot of it does come out of his pocket. But we all know what we're working for. We all know that we're going into the history books with this. So it's it gives you motivation that you wouldn't ordinarily have. If you
think that some huge university would just jump all over this and pay for everything.
Well, once we have it proven, then they'll be in line, hat in hand, please, please, please.
Yeah.
But until somebody takes that first step, and it's safe for them to do that, because if they tried to do it now, they get their heads chopped off by the conservatives out there,
right?
Conservative scientific peers who absolutely do not allow anyone in science to pretend that UFOs and aliens are real in any way. So it's just a psychological war that is waged by the conservatives in every field. I mean, just in every field. In all of life, there are those people who want to live in the future and those who want to live in the past. It's in politics, it's in government, it's in religion, it's everywhere. There are those who want to live in the future and those who want to live in the past. And, of course, I'm among the group that want to live in the future, and some scientists and some politicians and some religious leaders and all want to live in the future as well. But they are always overwhelmed and intimidated by the number and the virulence of those who want to live in the past.
I totally agree. Well, thanks, Lloyd. I appreciate you coming on, giving this update. Very important stuff going on. What's next?
Well, we take this information, this new data out, and we now start presenting that to investors, and we find the one who's going to say, you're right, this is really powerful enough. Let's go ahead and get it done, and then we'll be off to the races.
Okay, thank you. And keep me updated. Next time you have a big release, let me know. I'll put it out there, and we'll just let the people know what's going on, because I think the information needs to be out there on what you're doing. And more power to you, Lloyd.
Thank you very much. Pleasure to be with you. Talk to you again sometime.
Okay, have a good one. Bye-bye.